Hey there, Bio 20s. Mr. Beaudry here. Obviously, I'm not there today with you. In anticipation of not being there today, I recorded a lesson video tomorrow so that we are still scheduled for our quiz on Wednesday and we don't miss any important content. So, if you guys would do me a favor, please, and join me on this page in the notes, which is the Nephron that we drew yesterday. As you guys are doing that, I'm just going to talk about the quiz tomorrow, so please listen closely. The details are here. We're going to have a quiz. It'll be about 40 or so minutes in class on Wednesday, which will be tomorrow for everybody. Now, that quiz is going to be labeling a nephron. You'll have to show me where and what is secreted and reabsorbed. That part will make more sense after today as well. There will also be a couple of multiple choice questions and a short answer as well. Now, it's not going to be just on the nephron. This is going to be a chapter 9 quiz in general. It's going to be a low stakes assessment of what we've learned over the last 2 days. After today, ladies and gents, we are going to be done new content for chapter 9. It's going to be really short, and now we can see why we're going to pair that up with a major assessment with chapter 6 digestion. As a reminder, that is next Monday, so this coming Monday after the weekend, and we have our lab, our urinalysis lab, in the lab next door on Thursday of this week as well. Okay, now while we're here, hopefully we all see this page in front of us now. I should be able to see the nephron that we finished with in class yesterday. Now, a couple of parts I want to draw our attention to is some basic anatomy right now, just to refresh ourselves of the terms. First of all, I have my afferent arteriole that's feeding blood supply to my nephron. Now, the site of exchange here, of substance exchange, is called my glomerulus, and it is a dense capillary bed that's surrounded by something called my Bowman's capsule. My Bowman's capsule encompasses and encloses this glomerulus and encourages a pressure gradient that's going to shoot substances from my glomerulus into my nephron to begin filtration. Now, before we talk more about the nephron, I have my efferent arteriole coming out the end of my glomerulus after there's been a substance exchange to my nephron. I can see that it winds in and around my tubules, which is the next piece of anatomy that we can see. My proximal convoluted tubule, where reabsorption and secretion happens. Finally, through my loop of Henle, next into my distal convoluted tubule, and then it finally collects what's called urine in my collecting duct. My collecting duct is going to converge with other collecting ducts and form my two ureters, which feed into my urinary bladder. Now, please remember that once substances are inside my nephron to begin filtration, then it's not called urine yet. Remember that I call that filtrate until it gets finally to my collecting duct, and then that is called urine. That's when it's called urine. I also just want to make one more note as well before we move on, that my convoluted tubules are in what's called my renal cortex, which is the outermost part of my kidney. Now, part of my nephron does dip below lower into my kidney, and that portion is called my renal medulla. All right, let's move on here. This is just basic anatomy stuff that we just went through, so I'm not going to stay here. We're going to move on to this page right here. Yesterday, we ended by introducing the processes of the urinary system. All right, so we talked about anatomy of my excretory system, my kidneys, and my nephron specifically, but we didn't really talk about how they work or what I can call the physiology behind it. And then this is the physiology here, these 4 processes. Now, as a reminder, it begins with what I call filtration, and it ends with finally excretion. These processes in the middle, 2 and 3, reabsorption and secretion respectively, happen simultaneously, which means at the same time, inside various portions of my nephron, whether it's my tubules or my loop of Henle or even my collecting duct. There is reabsorption and secretion that happens in all 4 of those structures. That's what we're going to learn about today. Now, here's where we left off yesterday, talking about reabsorption, the second process. We talked in general terms just about amounts, relative amounts of fluid that are hitting my kidneys per minute, Finally, down to the very small amount right here that's actually becoming urine per minute. Remember, it's a very, very small percentage. What we should take from this relative volume here is that there is a huge amount of that volume flowing through my kidneys that gets reabsorbed back into my body. Remember that our bodies want to be as efficient as possible. We can reuptake, reabsorb, and almost recycle in a way what was considered quote unquote waste to begin with. My kidneys will reabsorb those back into my efferent arteriole. Now, here is going to be new content for us today, starting with this point right here. Okay? Now, my carrier molecules move sodium ions across the membranes of the cells that line the nephron. What does that mean? That means that sodium is going to be reabsorbed. And it's going to go from my nephron, it's going to go nephron to my efferent arteriole. Okay, and I'm just going to put one little thing, one little note here, which is going to be important. I can say that sodium drives the reabsorption of water. H2O. Okay, another way I can say that is that sodium is the driving force behind the reabsorption of water. And then that'll make more sense in just a few minutes here. So we've introduced my first substance that's getting reabsorbed from my nephron back into my bloodstream via the efferent arteriole. Now we're going to introduce something else. Now we have chloride ions and something that I can call bicarbonate. And bicarbonate is a basic substance. And bicarbonate is closely linked to pH balance in my blood, whether it's basic or acidic. It's going to be closely linked to that. And that's what we're going to end with today. Okay, now I'm going to go back up and add one more thing. This process where sodium gets reabsorbed is an active process. And we talked about that yesterday, active versus passive processes. This is going to require energy. So I have carrier molecules that are shuttling these sodium ions across back into my efferent arteriole. What's going to happen because of something that I call charge attraction, meaning that my opposite charges attract, the positive sodium ion right here wants to be closely linked to the negative calcium ion or the charge right there. So I will have chloride ions kind of flowing out of my nephron as well, passively. And same with bicarbonate, just because of this opposite charge attraction. This is a passive process. So if sodium leaves my nephron, then chloride and bicarbonate are very often going to just flow naturally with it and follow it wherever that sodium goes, in this case, back into my efferent arteriole. Okay, now moving on here, I have reabsorption occurs until the threshold level of a substance is reached. I've used this term, not threshold level, but I've used this term upper limit a few times with you guys. And again, there's an upper limit for everything in my body. It's not just going to reabsorb sodium, uh, chloride, bicarbonate, so on and so forth, water, to an infinite amount, right? There's internal mechanisms in my body that are governing the amounts that are being reabsorbed or being excreted as a waste. So whatever doesn't get reabsorbed, I can call that an excess of something. In this case, it's going to be sodium chloride. If an excess amount is going to remain in my nephron, then it's going to become excreted in the urine. Now, the same thing can happen with glucose and my amino acids. Remember yesterday that I talked to you guys about, supposed to be an arrow, about protein, my upper limit of protein. And most often we just urinate out excess protein because we can only have an upper limit of about 25 grams per meal, or between 60 and 70 grams per day. And again, these are substances that are not getting reabsorbed from my nephron back into my circulation. It's excess, so it's being excreted in my urine. Okay. Now, the last little bit that we're going to talk about is this portion right here. Now, solutes that are actively drawn out of the nephron create an osmotic gradient. And think back to up here where I talked about sodium drives the reabsorption of water. This is what I'm talking about right here. So, if I shuttle out sodium ions, if they leave my nephron, the outside of my nephron is going to be what I call hypertonic. And this is a term that hopefully we recognize from Science 10 or from Chemistry 20. What this means is that the outside of my nephron, more specifically my interstitial space, is hypertonic compared to the inside of my nephron. That means that there is an increased amount of sodium ions, concentration of sodium ions outside the nephron. Okay, the result of this, if I just make a bit more space here, the result of this is that water follows passively via osmosis. So again, always want to be at a state of equilibrium. If I have a really strong, dense concentration of sodium ions in one particular region of my body, in this case outside of my nephron, then water is going to flow across that gradient via osmosis and try and disperse the concentration of sodium ions. What it's trying to do is it's trying to lower the concentration of sodium ions, right? Not the amount of sodium ions, but the concentration. And that's why H2O, or water, is going to flow out of that nephron and become reabsorbed back into my circulation. Okay. Now, secretion is much more straightforward. There's less detail in general. So we're going to talk about secretion, and then we're going to get into another little bit of a diagram situation here. Secretion can be thought of as just the opposite of reabsorption. If you go back up here and think about reabsorption being the movement, here, why don't I just write it down right here? So reabsorption, forget about that, 3. Reabsorption is going to be movement from the nephron to my efferent arteriole. Secretion is going to be just the opposite. Movement of substances from the arteriole, now the one that's wound in between and all around my nephron, to my nephron. Okay. Now, what we have being secreted are going to be different than what's being reabsorbed. Remember that a couple of my big substances that get reabsorbed are water, sodium chloride, and then bicarbonate. Well, we have some different substances being secreted now that are going from my efferent arteriole back into my nephron. And those are nitrogen-containing wastes. So that's going to be things like urea. We have excess hydrogen ions, other minerals like potassium. Okay, these concentrations relatively are balanced by what I call secretion. Mhm. Now, typically, we know that filtrate that flows through my nephron eventually gets voided through my body as waste. So that's where these particular substances are going. This is stuff from my blood that I know I'm not going to need. Okay, whether it's complete waste and toxic to me, or it could be hydrogen, which is acidic. So, in this sense, it's not specifically a waste. I'm just trying to balance the pH in my body. This is going to be voided from my system because I cannot use it or I don't want to use it in this case. Finally, my fourth final process here is excretion, and then that just happens from the urine. Urine production is the final product of filtration, reabsorption, and secretion, right? Follows through my nephron, finally into my collecting duct, which is this unlabeled structure to the right that I see in this unlabeled nephron. And then it goes into my ureter, right and left, either one. And then those converge and join my urinary bladder, where it's finally voided through my system. Now, this diagram right here is an unlabeled nephron, very similar but not identical to the one that we just drew yesterday. I want you guys to take about 2 or 3 minutes here and draw this nephron fresh on a new sheet of paper. What I want you all to make note of here is that while I still have my glomerulus, I don't have my afferent arteriole. What I do have right here is my efferent arteriole. And I do just want you guys to draw it in a nice little line above my nephron, just for the sake of neatness and tidiness while we label this diagram in terms of substances that are reabsorbed and secreted. So go ahead and do that for me right now. Okay, I'm just going to add something here on the side as well. Make sure this is blue. Yep. Also, Bio20s, I am sorry if you can hear my dog sleeping and snoring beside me. Don't know if you can or not. Now, what we're going to add is a little list on the bottom left here that is filtrate components. And again, just make sure that we add this in as well. And essentially what we're going to do is we're going to tick these boxes off after we label them. So all my blood components that enter my glomerulus and thus my nephron, specifically that enter my nephron, are going to be things like water, sodium chloride, bicarbonate. I'm then going to have urea, glucose, amino acids. I have hydrogen ions and also potassium ions. And this is my comprehensive list of substances that get secreted or reabsorbed. We're going to add them to the correct spots in the nephron, specifically where in the nephron they get secreted or reabsorbed, and then differentiate in terms of whether it's being reabsorbed or secreted. And why don't we start with water? I'll give us just another 30 seconds to finish up that drawing. Okay, now we're going to start with water. Now, water is going to be very well reabsorbed in all of my structures here. So I can start with my PCT, or my proximal convoluted tubule. I have the reabsorption of H2O going from my nephron back into my efferent arteriole. Moving on, I also have the reabsorption of water in my descending loop of Henle. I have reabsorption of water here as well. And I also have water reabsorbed out of my collecting duct into my efferent arteriole. And also, please remember that my efferent arteriole does wind around my nephron, just for the sake of tidiness. We just have it up top as well. Okay. Excellent. Now, before we move on, remember that water here, I can see, and we will contrast it later to other substances near the end when we fill in this diagram, but water is absorbed almost everywhere in my nephron. And remember that we really want to try and reabsorb water, right? That's really going to mess us up if we have low levels of H2O in my body. So I want to do as much as I can to reabsorb water to my upper limit, whatever that might be. So H2O, perfect, got it done. Now let's assign sodium chloride to my diagram. Well, sodium chloride is getting reabsorbed. NaCl, and it's happening in my PCT. It's also happening in my my DCT, sodium chloride, and it's happening in a couple different spaces in my ascending loop of Henle. Here we go. There we go. A couple different spots for it right there. Now that's sodium chloride. Now please remember that sodium chloride actually happens in two different ways. It's not leaving my nephron as a compound. What's happening here is sodium is being actively transported out of my nephron back into my efferent arteriole. And because sodium is positive, chloride is negative, then I have that also chloride ion jumping in with sodium and leaving as a compound. Now, my last one here that's getting reabsorbed is bicarbonate. HCO3 is getting reabsorbed. HCO3 in both my proximal and my convoluted tubules. That's for bicarbonate. Again, which is my basic substance, which plays a role in the pH balance of my blood, is getting reabsorbed there. And again, that's because sodium is leaving as well. Bicarbonate negative anions want to jump in with that sodium and get shuttled across free of any energy expenditure as well. Perfect. Okay, now we're going to do urea. Now, urea is getting reabsorbed in my collecting duct. Oh, you know what? We actually missed one more area for sodium chloride as well. So sodium chloride and urea are both getting reabsorbed out of my collecting duct as well. So I can have urea being excreted because it gets secreted back into my nephron from my blood, but there is an instance, as it is right here, where urea gets reabsorbed as well. Okay, urea, perfect. Now, the last couple substances that are getting reabsorbed, and we can see from this list of filtrate components that most of this is reabsorption as opposed to secretion. We haven't talked about secretion yet. Now, glucose and amino acids, we're going to group them together and just say that these are nutrients. Just to save a little bit of space, and I'm talking about glucose and amino acids. The reabsorption of nutrients happens in my proximal convoluted tubule. So there we go. Reabsorption, reabsorption. So if I take a look at all of these substances right here, I can see that water gets reabsorbed almost everywhere. Sodium chloride gets reabsorbed almost everywhere, but nutrients only get reabsorbed in my PCT. I can contrast that with bicarbonate as well, both my PCT and my DCT. So I can say that most of the reabsorption happens in my PCT. Okay, the vast amount of substances that enter my Bowman's capsule get reabsorbed in the PCT, the amount of substances in terms of the different types, but also the general amount of reabsorption that happens as well. relative to other areas happens in my PCT as well. Now we're going to talk about secretion, and we're just going to change my little color here. Perfect. Why don't we do green instead? And green is going to represent secretion. Let's do hydrogen first. Okay, hydrogen is going to get secreted from my PCT as well as my DCT. And again, the reason that I want to secrete hydrogen ions from my circulation into my nephron to get excreted as waste is pH balance. If I have an acidic blood level, too much acid in my blood, then I'm going to want to decrease that acidity somehow. Acidity is closely related to the hydrogen ion concentration. So if I get rid of my hydrogen ions, it makes sense that I'm going to lower my blood pH. Now, the last substance on there is potassium ions. Potassium ions are getting secreted from my efferent arteriole in my DCT. So let's stop here and take a little glance at what we just did. We've labeled another nephron, but we've labeled it differently. Now we've talked about processes of excretion, not just anatomy. All right, this is what happens in terms of reabsorption and secretion, processes 2 and 3 respectively in each part of my nephron. Now, this is what the quiz should look a little bit like. We need to know where these substances get reabsorbed and secreted. Now, it can get confusing in the sense that I'm doubling, tripling, quadrupling up in some cases, like NaCl and water, in terms of the locations that they get reabsorbed from. So just be aware that we need to take care to keep these substances straight. Let's move on. Here is a very brief snapshot of a summary of what we just did. If it's right here, why did we just do it as a class? Maybe you're frustrated, I'm sorry. However, this is going to be by far the better way to learn this stuff, rather than just staring at a diagram that's been taken from a textbook. Okay? If we take the time to do this, while we explain it as we go through it, this is being retained much more than taking a glance at this thing for a couple minutes and relying on that. Okay, we're getting close to the end, ladies and gents. We're doing well. Now, water balance. I've mentioned a little bit about water balance, but mostly about pH balance, which is what we're actually ending on today. So let's talk about water balance right now. Let's just see, still on green. That's okay. Now, my kidneys are going to adjust for increased water. If I increase my water intake goes up. Then my urine output is also going to go up. And that just makes sense. It's a direct relationship when we think about it like that. If I drink a lot of water, I'm going to be overhydrated. I'm not going to need as much reabsorption through my nephron. So I'm going to leave that H2O left in the nephron. H2O gets left in the nephron, which means that there's a decreased reabsorption. Now, on the other hand, if I have a decreased water intake, then urine output will also decrease. And that's because I'm going to have increased H2O reabsorption. If my systemic level of water is low, then my nephrons are going to recycle and reuptake that H2O, bring it back into my efferent arteriole from those various parts of my nephron. Okay. Now, by talking about these two different statements, I can see here that my kidneys are highly involved in regulating body fluid levels, not just my water balance, but also blood levels as well. So how we do this, there's a couple different mechanisms, and it typically is a relationship between my nervous system and my endocrine system. Again, when you guys think endocrine system, I want you to think hormones. So, yes, there are a couple of different hormones that we need to know that help regulate my water balance in my body. Don't worry, it's not nearly as many hormones as digestive. There's only two that I want you guys to know. The first one is what I call ADH or antidiuretic hormone. First of all, let's break this down. Anti being the first term there, diuretic being the second. A diuretic, for those of us that don't know, is, let me erase this. A diuretic is something that makes you pee. Very simply put, something that makes you increase urination or urine production. If I increase urine production, then I'm going to be urinating more. So, a couple well known diuretics are going to be caffeine, and a huge one is alcohol. So if you drink a lot of coffee or if you drink a lot of alcohol, you are going to be urinating very, very frequently because your urine production is going to go through the roof, especially with alcohol. It's going to absolutely skyrocket. So, knowing now what a diuretic is, the definition of a diuretic, think about now the hormone, the name of the hormone. It's very self explanatory. It's an antidiuretic hormone. Let's jump down to the end product first. Here we go. The end result is that I have a decreased urine output. If I think about the name of the hormone and compare that to what a diuretic is, that makes sense. An antidiuretic is going to be something that's going to decrease urine output, and I'm going to be urinating less frequently. So, how does ADH work? It's going to act on the kidneys to increase water reabsorption in the nephron. Increase water reabsorption in my nephron from my nephron, however you want to say it, back into my efferent arteriole. So we will be able to contrast that with my second hormone. And the very, very important thing to know is that it increases water reabsorption among all of those substances that we talked about being reabsorbed and secreted, right? Sodium chloride, bicarbonate, so on and so forth, other nutrients like amino acids and glucose, it's going to increase H2O reabsorption. Okay. So the second kind of hormone that's going to help regulate my fluids is called aldosterone. And aldosterone is not an antagonistic hormone like we talked about insulin and glucagon being, right? Those two are antagonistic to each other, meaning they have an opposite effect for my digestive chapter. Aldosterone is going to have the same end result as ADH. If I look at ADH's end result, let's go back up here, it decreases urine output. If I look at aldosterone's end result, it also decreases urine output. But the mechanism behind how is what's different. First of all, aldosterone is a hormone produced in the adrenal cortex, outer region of my kidney. There it is right there. All right. And it's going to act in response to a drop in blood pressure. Drop in blood pressure. This also means that I have a drop in blood volume. If I have a drop in blood pressure, that means that I have less blood, right? Maybe not the formed elements. So, not my blood cells. That count still stays the same. But my plasma portion of my blood, which is mostly water, is going to be decreased. So, this is what I'm talking about when I say the mechanism behind aldosterone is different than ADH. It's acting on nephrons to increase not H2O reabsorption, but sodium reabsorption. This number is incorrect. Ooh, there we go. Sodium reabsorption as opposed to H2O reabsorption. Please remember that sodium is my driving force. So think of it this way. As sodium chloride is reabsorbed, this is a salt. Reabsorption increases, more water will move out of the nephron by osmosis. So if NaCl reabsorption increases, again, my interstitial space is becoming hypertonic. Relative to my nephron, I have a higher concentration of salt particles outside of my nephron compared to inside. So water is going to move out passively via osmosis to try and cancel out and redistribute those sodium chloride particles. The end product is this: I have a decreased urine output because I have more reabsorption happening. Decreased urine output, increased reabsorption. And we can add that to this one as well. Decreased urine output, thus increased reabsorption. Okay, ladies and gents, last slide. Doing good. The last little bit that we have to know for chapter 9 is this: my kidneys play a role in maintaining blood pH. Remember, we talked about a substance being basic or acidic. Well, my blood is the same thing. My blood has to be around a pH of about 7.4. And please remember from chapter 6, digestion, that my cells and specifically my enzymes from chapter 6, they have to exist in a very neutral state. It's only those stomach enzymes that thrive in a very acidic environment, very, very low. So if I'm too basic, not good. If I'm too acidic, not good. How do I regulate this acidity or this pH in general? Well, it has to do with my kidneys. And this buffer system is going to resist changes in pH by adding or removing hydrogen ions and also CO3 negative, which again is bicarbonate. So situation number 1, if my blood is too acidic, then hydrogen ions are going to be excreted, which means that these will be secreted by my efferent arteriole back into my nephron, where it can join the urine. Hydrogen ions can join the urine, and I can void it, excrete it. Now, conversely, if I get rid of hydrogen ions, that's going to increase my pH to more basic. If I, at the same time, reabsorb more bicarbonate, remember that this is basic in nature, that's going to raise my pH as well. And raising my pH is going to bring me back to a more neutral state if I'm too acidic to begin with. Situation number 2, if my blood is too basic, just the opposite is going to happen. This hydrogen is going to be retained. It's going to be retained, and I can say that it's not going to be secreted by my efferent arteriole. It's going to be, it's going to stay in my systemic circulation. And then also, I am going to have my bicarbonate. It's not going to be reabsorbed. This is going to be excreted, right? Not secreted, because this is in my nephron to begin with. If something is being secreted, it's going from my efferent arteriole into my nephron again, but it's not. This is already entering my Bowman's capsule. So it's just staying in my nephron, and eventually it's going to be excreted, just like that. Okay. Now, that's it. That's what we have for chapter 9. Congratulations on making it through. Very proud of all of you. Hopefully, we understood this. Again, I'm sorry that I couldn't be there with you guys today, but I'm happy to answer your questions before the quiz tomorrow. If we have any kind of questions like that at all, I will be available all of the day by email from home, and maybe some of the evening as well. But if you guys have questions, and please trying your best to get them to me during the school day or shortly after it ends by like 5 or so. Okay. So right now, you guys should be spending the rest of the class working on the urine analysis case study, which again will be due on Friday of this week. Again, it's quite straightforward. You might have to do a little bit of additional research on your phone. Just to answer question number 1, only number 1. Let me say that, only number 1. After that, This is going to be questions. Content from our notes will help you answer. We shouldn't have to do any additional research at all. This case study is going to prepare us very well for the quiz on Wednesday as well. So I would say it's in your best interest to work through that and answer those questions in class, or maybe even this evening when we go home in preparation for the test tomorrow. Okay, that's all from me. I'm going to say goodbye, and I will see you in class on Wednesday, hopefully. See you later.